I would just like to discuss very warm welcome from my side also to all of you. Well, as already the committee has introduced me that okay, since last 23 years I am working for biotech, especially Ploidy breeding. So I would just like to put some focus on to accelerate agro industry via flow cytometry. Right. During my journey, I have worked for so many crops like chili, capsicum, cucumber, broccoli, rice, so many, mustard. So you name the crop, I have worked for that, and I have gone through for melon, watermelon, cucumber. I'll share my experience using flow cytometry. Fortunately, since 2000, I'm using flow cytometry. So I think it would be good for me if you can make use of my knowledge and my work what I have done. Let's continue. We can foresee that okay, what would be the upcoming challenges for our breeding. So we would require, I would say, biofortification means nutritionally rich products would get required. Then fast replacement of the existing product, which is like I can say that because I'm working for commercials, then our all the germplasm is getting sterile, environment is getting shift, and new races of pests and bacteria and viruses are coming. You can see that okay, heat waves are there in the one of the picture. Then population is increasing so fast, and like you can see that okay, garbage everywhere, then drought and lot of pests. Then we are making concrete jungle for our living being. So these all are the upcoming challenges. And of course, like to live all the people, we are going for deforestation. So these are all the upcoming challenges, which is also I would say ongoing. So to overcome these one, we have to remain prepared. In my opinion, these would be the future requirement from our hybrids or like varieties what we release in the market. We would require disease resistance. We would require better process quality and seed production. We would require products having a better shelf life and better nutritional quality. We would require higher yield. Of course, it is the prime requirement by all the farmers. And stress resistance is also very much requirement. Then photo insensitivity and adaptability would get required in the hybrids. To achieve that, there are several breeding methods. So we all know that okay, conventionally, first we go for the identification of germplasm, then we go for pure line selection, then mass selection, pedigree method, single seed descent, then back crossing, then recurrent selection, synthetic seeds, heterosis breeding. But all dependent on hit and trial method. So there are some non-conventional methods also, like genetic engineering. It's a long-term known process now. Marker-assisted selection is MAS. Tissue culture is pretty much important, play a role in the breeding. Then somatic hybridization, and of course like mutation breeding as well. I would say that okay, there are some advanced breeding techniques like polyploidy breeding, for which we are going to discuss, and the Sysmex presentation you might have here and listen that. Then of course gene editing is the latest one. Then how you are going to get your designer crops. That means you design the crop. You say me the okay, these are the particular traits which I am looking for in my hybrids, and we can try to get that. We can design crosses to get minimum number of crosses, and from those crosses we can select that what we were requiring. And of course the speed breeding, because that would also the advanced breeding techniques which helps breeders to get the best products. For any conventional breeding methods, the requirement is inbred line development. To get that, I would always focus since 2000 that okay, you require doubled haploids. This is nothing but it's a pre-breeding tool, because for any hybrid development, if you like to get completely homozygous inbred line, so that could get solved getting doubled haploids. So I'm going to put focus. So if you have any queries, any questions, any doubts, you can put that okay cumulative form, and I can try to answer all of them. Well, just look this slide. Here is one like blonde male and blonde female, and all these five kids are their own. Well, their own. I mean to say that. Is it possible? Think like that. Is it possible? I would say yes. The black baby from these two blonde guys is also possible. If like any of the forefather from the mother or father, if that was having some black blood, means the blood from the countries having black baby or black skin, so that is possible that in a certain generation it gets in the homozygous condition, they get like recombinant and they get a kid having dark skin, gray skin. So this is possible. Now come to the crops. If we discuss about hybrids, you see the cauliflower example, the female and male, you get cross and you get like the crop. I would say you get crop either like 5 plants, 10 plants, plants in 5 acre, 5 hectares, and 5 villages, 5 cities, 10 cities, 5 countries. If it is homozygous, then your hybrid will remain uniform. So, well, you get like fixed parents, and from those fixed parents, you get from the fixed hybrids also. Well, I would put some focus on this double haploidy. What exactly is double haploidy? This is a tool for true parental line development, I would say, in bread factory. So already, like you, most of you know that the term haploid refers to those plants that possesses a single set of chromosomes in their sporophyte, because sporophyte normally is 2n. So haploids are an individual with the gametic chromosome in its somatic cells. I mean to say somatic cells, because during like gamete formation, meiosis occurs and chromosome number gets half. In example, like 2n number of cabbage is 18, in chili or hot pepper it is 24. So this is something like that. So doubled haploid, an individual with a double number of chromosomes. Well, in case of triploids, there are 3 sets. Tetraploids, there are 4 sets. In polyploids, there are multiple sets of chromosomes are available. Well, I would say that okay, how we get that double haploids. You see this diagram. In this diagram, the blue portion, where like okay, after meiosis we get gametes, male and female gametes. So before fertilization, they remain in haploid. So during that time, if you capture any of the gamete and from the plant, so that remains haploid. I can see you, like I can show you. This is better in this picture. You see this particular diagram. So after meiosis, you get like microspores and megaspores. Then megaspores form eggs, microspores form pollen. After fertilization, you get like a fruit. In that fruit, you get seeds and like a complete plant cycle. Uh, the below the red line, when it is in the gametic form, in the haploid form, if you get like develop a plant from that, it is always a haploid, which gets doubled using diploidization treatment, which we will discuss even during this presentation itself. We get double haploids. So this is the flow chart of getting DH lines. There are different methods of producing haploids. So most common and popular is androgenesis, whether it is through anther or microspores, or even I would say irradiation of pollens also. Then gynogenesis means using ovules and ovary culture. You get like double haploids. Then delayed pollination is also a particular technique. Then chromosome elimination, distant hybridization, which is in use since long time, and use of inducer line. So I would say that in using inducer line, there are two different latest methods. One is Senesh3, which is, I would say, even not that much latest, because it has been popular and paper has been presented by Ravi and Chan for Arabidopsis, and like in some of the brassicas now they are trying. But now after getting gene editing, so using gene edited inducer lines, you can get even haploids, but the still the frequency is pretty low. But I would say this is future. So the basic mantra of getting double haploids, different methods, you have to culture haploid cells only. Well, this picture is from my own lab. So you can see in the center donor, you can see that, okay, this was the chili. And once you get like DH population, you see that, okay, there are some thin chili. You can see that some capsicum kind of chili, bell pepper kind of chili, then blocky kind of chili. So there are so many variants, though so many recombinants, which remains gets in the fixed form. So this is the beauty. Now you see this example of cauliflower. So well, all these pictures are from my own lab. So you see that, okay, if like some kind of disease infestation was there, so in the center that all DH lines, all plants are diseased. Even like you see any picture, it is so uniform at each and every locus. The crop remain homozygous in homozygous condition. Well, this is the basic picture you will which you can get from the net. I would just like to put a lot of focus on this slide because this tells seven lot of things. You see this slide, it tells that, okay, DH technology versus conventional breeding. So I have taken here two characters only which dependent on A and B, capital A, capital B, small a, small b. So using conventional method, you get F1, you go for selection, go for F2, F3, F4, F5, F6, F7. So much variation you go forward. But like, okay, once you get like final, so even then you get like, okay, we say that, okay, 100% homozygosity is there. But I would say this is asymptomatic because like on papers it is possible, but in field once you get that, it is, I would say, that arbitrary, because like our selection is based on phenotype, which depends on the genetics and environmental factor. And in the farmers field, environmental characters or factors are not in our hand. Whereas in terms of DS technique, you get like gametes. If you get like capital A, capital B, after chromosome doubling, you get like capital A, capital A, capital B, capital B. Since similarly, like small a, small b, capital A, small b, whatever like gamete condition was there, you get like exact double of that. So this is the Punnett square, which we have studied during our basic studies. So you see that out of these 16, if like this is the allele information, all these plants would look alike. So after like getting these much number of plants, which like when I am taking example of only A and B, so this is the picture that out of these, because these all are alike, how you are going to select that? If like, okay, it is the example of A, B and C, the picture is even more bit more critical. So all these plants would look alike, so selection of one plant or two plant out of these is like pretty difficult. I would say like first plant, which is like capital A, capital A, capital B, capital B, capital C, capital C, if you are going to select that, it is very difficult to select this plant. So even like I would say this is again like when it was only A, B and C. Now you think about one of the trait which depends that, okay, on multiple genes A, B, C, D, E. So if like this is the condition, then it is seriously difficult. And once like, okay, you see even right side, so when like none of the allele is dominant, then this is the situation. And when like, okay, only three gene are dominant, whether other three in B are dominant, so situation would be entirely different. So from these much number of plants, if you like to select only one or two, it is sincerely difficult. Let me tell you, it is sincerely difficult because environment is not in our hands in the farmers field. So to get like such kind of situation, we have to be very cautious and we have to be very careful that, okay, once we go for like DH development, we get like real benefits. So what happens then? What is the real situation? We get like phenotype, which is dependent on G into E. If any of these two changes, phenotype changes. Reason to breeder, not known. G of plant, not known. Could be easier for monohybrid process where characters are governed by single gene only. But if traits are polygenic, as I already told, it is sincerely difficult to get the parents in the fixed form conventionally. So what happens? So possibility of selection of a line which is not 100% homozygous, it happens. Reason, it could be due to cross pollination. Mostly it could be due to environmental condition, long duration crops, unfavorable time of evaluation, and many more. So, for a commercial company, I would say what happens for 1, 2, or 3 generations when like demand of seed is in lesser quantity, hybrid performs well, and when this heterozygosity accumulates, then variation get observed in the field, and you get like several complaints from the farmers. This is like again a chart which you can get even on net also. It is extremely effective in combination of multiple alleles, especially recessive genes. So you see that okay, when we are working on single loci in the conventional method, out of 4, one could be like of having your trait, whereas in DH it could be like 50%, out of 2, one could be of your trait, target trait possibility. So similarly, like 2, 4, you see the example of 4 loci. If you are focusing, then out of 256, there is only single plant having your traits of interest. Whereas in DH population, out of 16 plants, you will getting like a single plant of your having trait of your interest. So if it is like even because like most of the disease resistant traits are multiple, dependent on multiple genes. So QTLs. So if it is like dependent on 10 loci, that okay, you see that okay, out of that much number, so 100,000, so you get only like single plant, and in DH 1024 you get like probability that okay, you get single plant. I am telling about like capital A, capital B, capital C, means all genes are in capital form. So what is the practical situation? Very few breeders plant only F2 population of more than 1,000 plant, and 3 to 4 replication at different location, and use support of technology also markers. So this is why, like, because everything the selection based on breeders eye, I would say that this technology is pretty useful. So what are the different challenges with DH production? Generally, the starting setup is bit expensive, I would say. Very important is expertise of team is required and facility is required. So it is dependent on restriction of number of crosses. This is the basic problem that okay, all the most of the tissue culture scientists, this technology gets proved under lab conditions, because scientists are show their very much happiness showing their product in the lab. They say that okay, this is my box, this is like single plant, I got this plant, they published paper. But like real situation is something different, because like if technology would not get proven in the field, then it's of no use from my side. And like donor selection, this is also most important to get like this technology. And most important after that, if you get like DH line, use of that DH line without confirmation. So what are the specific things which are specific challenges? Of course, this technology is genotype dependent, lesser regeneration frequency of haploids, standardization of protocol is required for different plants and species, donor material care, and reduces genetic diversity. This is the basic statement, because like it doesn't reduce the genetic diversity, actually it fixes the genetic diversity. After DH, as we already discussed that okay, polyploids, because these are also the game changer technology for future. So what are the polyploids? So polyploids, the condition in which a normal diploid cells 2n or organism acquire one or more additional sets of chromosomes, they get like polyploid. So how you go for like polyploid identification? Either if changes in chromosome number, so it may occur that okay, cell size also get changed, stomata size may get changed, flower size may get changed, flowering time may get changed, fruit size, and like many more. uh, this I think a basic example you must have seen during your studies time. This is the U triangle of brassicas. You see that okay, A, B and C combination, this Brassica campestris, where 2n is equal to 20, it's Chinese cabbage or turnip wrap. Then in when you see like capital B, Brassica nigra, this is black mustard, where like n is equal to 18. And like Brassica oleracea, C genome, where which is like cabbage, cauliflower, broccoli, kale, kohlrabi, and Brussels sprout, where n is equal to 9. So naturally it occurs in the nature, and you see that they all are brassicas having same number of chromosome, like bit different, and even see how much difference is there in cabbage and cauliflower and broccoli and kale. See, this is the beauty of nature. Now, if you form like crosses between A and C, A and B, B and C, so we get like okay, Carinata, Nepus, Gentian, so much examples. So this U triangle you must have seen. I will show you that okay how you can use the flow cytometry to identify even like these different species as well. So I would say that okay, Ploidy breeding could be the game changer in the near future. So what are the different ways to modify ploidy? I would say using seed treatment, embryo treatment, root dip treatment, auxiliary bud treatment, callus treatment. So many options are there, so many. And so many chemicals are also known, whether it is colchicine, oryzaline, GA3, caffeine, trifluorelin, APM, even I would say like UV radiation or even like X-ray, cobalt. So many things are there which can change like ploidy. So what are the general characters of polyploids? So triploids, tetraploids, hexaploids, they are generally vigorous in nature. Generally, it is not common, but they are generally vigorous in nature. Polyploids often thrive better and grow healthier. So interspecific hybrids could be another option. And triploids generally, usually I would say they are seedless. We know like examples of cucumber, goats, tomatoes, eggplant, kinnu, oranges, watermelon, banana. They are like triploids are seedless. Then what's required? Well, selection. If like okay during selection we are not getting any aneuploids, and the stability of polyploids, which is like most important. Here are some examples. You can see that okay now in the market grapes are of very big size. So these grapes are not like normal grapes which we were using 20 years back. That time we were using only diploid grapes. Now we have like tetraploid grapes. So we can see like okay the difference in the size. If we see the leaves of watermelon, tetraploid watermelon, you see like okay the size of the leaf is pretty big as compared to diploid. So these are again like how you get these tetraploids, triploids also. If you have like tetraploid seed parent, I would say diploid egg, because like gamete would be diploid, and you get like a male plant, two and pollen plant having haploid pollen, you get like triploids. So well, personally, I'm disclosing that okay now we have developed seedless capsicum also, that is triploid. In the bottom you see that okay how you get like watermelon triploids. So again the same example and same way that okay four and female parent and two and male parent, you get like a triploid watermelon having almost nil seed or like ill developed seed, I would say. Well, that was like till now we were discussing only about vegetables and other crops. Polyploids have very good future, which I personally see in floriculture. You look for like dianthus, you look for jasmine, you look for anthurium, you look for gerbera. So we get like we have customers which we look like intense color or size or number of whorls, intense color, then fragrance. If we go for like polyploid formation, so we get like any of well we can even like target our genes also, that okay which particular gene we are looking. Otherwise I in naturally and normally if we go for tetraploids, so we get like a drastic change in their morphology, or even like product also, metabolic product also. So whether it is fragrance or like if you are we are thinking to extract some essential oil from out of them for perfumery, so this could be like very good example. So in the market some known polyploids I have mentioned here, like sugarcane, which is normally ATEX, coffee, which is like diploid also, tetraploid, hexaploid. 20 years back even like cotton was also diploid, but now all the cotton mostly cotton is tetraploid now. So sorry, brassicas, then potato, we know all the potatoes are tetraploids. Strawberry, this is wonderful example in the market. It is like 2 and 4 and 6x diploid, tetraploid, hexaploids, octaploids, because like this is beautiful crop and you love the fragrance, you love the size, you love the taste. So from ploidy we have changed that. Peanuts are also tetraploids, oats are also hexaploids, and bread wheat is also hexaploid. So these are like different examples which already polyploids are available. So this is another example of Ophioglossum. In like, okay, you see the like difference in the leaves of Brassica oleracea, Napus, and Rapa, and even like flower size also. You get like change, and you can identify. Now it's a question for you that okay, you can think for polyploid for metabolites, because if you are going to extract some specific chemical from these plants, so you can get like better products, or like intense products, and you get like better quantity also. The chemicals which are used in drug industry, you can extract that. Then oil extraction, you can think about jatropha and other things. You can think for woody species, because the wood is absolutely required. And if you get like polyploids trees, so wood quality would also be much better, and even like quantity as well as quality. Fragrance, already we discussed that okay, you can think about the chemicals, and which can used in perfumery, and many more I would say. What is required? So protocol for ploidy alteration, yes. Confirmation for ploidy, evaluation in field, then after evaluation, hybrid formation for commercial release or direct variety release. Now the question comes, how to confirm ploidy, which is our like utmost question. Qualitative as well as quantitative. I would say chromosome counting. All of you would get agree with me. Chromosome counting, counting is the best possible way. My question: how much plants you can count for chromosome in a day? 1, 2, 5, 10, 20. That's it. Not more than that nowadays. If I am thinking for like 100, 200, 500, thousands, so I have to go some method. So accuracy should be there, and cost should be very much effective for ploidy analysis. Here comes the role of flow cytometer. This is like flow cytometer which I am using in our lab. So I would say how you are going to use that. Ploidy is the number of sets of chromosome in a cell. So flow cytometry, the prevailing method for the measurement of nuclear DNA in plants. We are discussing about plants only. So samples are typically prepared from only a few tens of milligrams of plant tissues. It facilitates the analysis of samples with asynchronously dividing cells. I would say like mitotic active cells are required for ploidy analysis, and high precision with coefficient of variation is pretty much required to get best DNA peaks, which ranges from 1 to 5. Usually we get like 3%. So this picture is how you prepare sample. We get like okay, small plant leaf samples. We cut that, and during cutting you won't believe that okay, my technicians are so practical now. They go for chop 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 chop, means 10 to 16. They are so practical. They take like very less time in forming homogeneous samples. After that sample you go for like okay, filtration to get like DNA and go for the analysis. So it is like pretty easy and pretty fast also to get like a ploidy analysis done by using flow cytometer. Especially I'm talking about Sysmex. So this is the normal cell cycle. For the mitosis, we get like synthesis phase, then G1 phase, synthesis phase, G2 phase, and mitotic phase. Once we go for the analysis, we get like G0 and G1 peak, and synthesis phase, and like G2 peak. Usually in our analysis, we get like 2 peaks. Now I'll show you this is the diagram, the pictorial diagram, what actually happens in this flow cytometer. In this flow cytometer, in this DNA, all the cells, this machine tries to put that in a particular beam, particular line, that okay, that in that cuvette single cell passes in front of the laser beam, and when that laser forms on that, so we get like okay, reflection, and after reflection, so many detectors are there in that flow cytometer, which detects and give the result. So this is the fluorescence emission, and form that okay, how this particular cell goes, and after fluorescence we get like picture. So there are two fluorescence detectors are there, orange and red, but that also depends that okay, which particular dye you are using. Like I'm using DAPI. So for DAPI, I require only UV light. So that is there. Two different kind of plots we get from Sysmex flow cytometer. So one is like single color histogram, and even like two color dot plots also. It also depends that okay, what exactly you are looking for. Now these all are the like ideal pictures. I'll share some pictures of my own lab. So this is also like okay, if you get like a picture in this format, we see that okay, where the peak is rising, that okay, if like haploid peak is there, or diploid is there, or triploid is there, tetraploid, hexaploids, even like a cDNA content is also pretty much known using this particular machine. So I would say for my 23 years career, it is very useful. This picture is from my own lab. You see at the top, this is the control. The first peak is noise because the sample was not very good. So noise peak is too high, which shouldn't be there. But this is the practical thing which I have put this picture, because once we get that, we think that, okay, this is the G1 peak. No, this is the noise peak. Second big peak is the G1 peak, and the small peak is G2 peak. This is the control. Now from the samples, if haploid is there, this sample was preparation was very good. You see that okay, noise peak is pretty small in size. Then G1 peak is pretty big, very good peak. G2 is also a good peak. You see that okay, the position of these two peaks have got shifted towards left side, exactly at the half. So we say that okay, it's the haploid one. So in the second one, we see that okay, the DH plant population, the sample was very good. So this peak the noise was very less. Then G1 and G2 are pretty sharp. Because we have developed this from microspore, so we say it is DH, and there is because it tells about ploidy, not that okay, exactly that okay, the diploid and double haploid it cannot distinguish. In triploid, you see that okay, these two peaks G1 and G2, they have got shifted towards right side. In tetraploid, if you see the first picture of control picture and compare with this one, you see that okay, this G1 and G2, they have got shifted towards right side, exactly towards double. So just by seeing the peaks, we can say that okay, in comparison to the control, this is the double. So if like control was 2n, this is the 4n, then 6x. Now at the last, you see the aneuploid. I would say this is like okay, whether it could be 2n minus 1 or 2n minus 2. Reason being the plant peak has been shifted towards left side. If it was like bit towards right side, then I was able to say it could be like 2n plus 1 or plus 2, something like that. If exactly we like to know whether it is minus 1 or minus 2, we have to do some more exercise and we can easily get to know. So what is the role of flow cytometer in R&D and production? Well, as already we discussed quite a lot that okay, in R&D directly we can use this flow cytometer in breeding, whether it is for haploid development, double haploid development, for pre-breeding exercise, we can use that. If we are transferring our traits from wild species having different number of chromosomes, it is a very good device, I would say. Because like okay, if you are having like a chromosome number of a variety is different and 2 is different, once you mix that up in that cross, in that hybrid, if you will check the ploidy, it the in the first one the peak would be at one place, in the second one peak would be a different place, in the hybrid that peak would be at the intermediate level, which will confirm that okay, you get like okay, your hybrid of both the entries and your trait is getting transferred, right? At least you got like hybrid formation. So in production, it is basically used when related species are lying in the nearby production areas like brassicas and okra. So we can go for like random bulk analysis of parents, which tells that okay, if like some kind of mixture is there, so we can easily get to know. Likewise, parents, we can go for the random bulk analysis of hybrids using flow cytometry, and we can easily get to know. So if like okay, any tetraploids or any polyploids, these are under production, so the flow cytometer can easily tell us. And for the production, if we get like okay, different yields from different areas, it also depends that okay, whether the pollen was viable or not. So it also tells that okay, the viability of pollen and the flow cytometer could pretty much helpful. So as I already told you that okay, this slide I have refer from the Professor Delizel. So this is like detection of somatic hybrids. How is it possible using flow cytometer? Again, I told that okay, if we if we label one of the cells from one entry with some fluorescent dye and another entry with some different fluorescence dye, we make some crosses during our exercise and analysis using flow cytometer. In the hybrids, we get like fluorescence of both the cells, and we can easily get to know that okay, the somatic hybrid got formed or not. So this picture you might have seen with the molecular markers only, right? This is also possible using flow cytometer. The condition only. So if we are going for clustering on the basis of nuclear DNA content, please be sure clustering on the basis of nuclear DNA content, then of course using flow cytometer we can get this kind of cluster. We can get like okay, these are that from different species, different plants are there, and we can easily get to know that. So I would say thank you so much to hear me. And well, my experience is always there for all of you if you have any questions, because like okay, I would say that okay, now we are working for gene editing and designer crops, and we are using intense use of flow cytometer, and I have used like flow cytometer from different companies, but the kind of accuracy, the kind of cost effectiveness what I have seen from Sysmex, earlier Partech, I have never used that from any other companies. Truly speaking, because I'm not from Sysmex company, I'm not a Sysmex person. So I'm a scientist, so I'm working for my crop improvement. So, but thank you, Sysmex. Thank you so much. And your system is really good. I would love to use that forever. Thank you.
Yeah, so I will talk about I have 5 agenda points. I give a short introduction to Ploidy analysis, and I will speak a bit about laboratory experience and practice, how it works, how to prepare samples, and give some tips. I'm presenting the components of our Sysmex solution, and I will show some exemplary applications. And I will give some words about benefits in breeding and research of the Ploidy solutions we have. Okay, let's start. Okay, when we talk about Ploidy, in general, Ploidy concerns the DNA content of an organism. So usually I'm speaking now about plants or animals. So leave out bacteria or fungi. But usually, well, the chromosomes are those cell components which are made of DNA, and they bear the major information of the major genetic information. And chromosomes are formed by 2 chromatids, which are practically identical, and they are present in different numbers in a specific species. So, for example, if we look at us, at humans, we have 23 pairs of homologous chromosomes. So homologous means they are very similar, at least. They have the same structure and size, and mainly also the same content. So we have 23 different chromosomes and 2 of each. So that means we have in total 46 chromosomes. Yeah, and here we are already at the definition of what ploidy means. If we have two sets of homologous chromosomes, we call such organisms diploid. Two sets, diploid. Of course, that in a normal generation cycle, one of the chromosome is coming from the mother, and one of the homologous chromosomes is derived from the father. If we look at animals or mammals in particular, practically all are diploid. There are very few variations. If we look now into plants, the situation is pretty much the same. So plants have also usually a certain number of chromosomes and two copies of each of these chromosomes. So that means most of the plants in the generation cycle of higher plants are diploid. Another term which can be of relevance, we have to discriminate quite well, is the genome size. So the genome size is related to the absolute quantity of DNA per cell or per nucleus. So this is not counted in sets of chromosomes, but in picogram DNA or in the number of base pairs. So that's the total amount of DNA, and when we speak about genome size, we have to also define on what level of ploidy it is referring. So usually it's referred to the normal single set of chromosomes. So then we call it the 1C value of genome size. Okay. Good. You can see in this figure here examples, let's say, of different number of chromosomes in one plant. A classical method of ploidy determination is to take the meristeme from root tips and to prepare that in a microscopic preparation, and then you are able to see the chromosomes when you have a nice preparation, and then you can count that. Later in flow cytometry, we are doing that different, but we will get such figures here called DNA histograms, and then we can assign different levels of ploidy to certain numbers of chromosomes in one particular plant species, for example. Okay, so as I said, ploidy is defined as the number of homologous copies of sets of chromosomes. So we can have in plants, and this can be a natural process, but it can also be an induced process by breeding strategies, breeding processes. So the regular level would be diploid. But of course, when there is a artificial modification of the ploidy by breeders or breeding processes, then triploid plants can be produced which have 3 sets. Tetraploid plants can be produced which have 4 sets of the same chromosomes, or we speak about polyploidy when there's more than 4 sets, so 6, 8, 12 sets, whatever. There are different higher levels of ploidy, of course, thinkable. Now, some exemplary genome sizes in contrast. So we see here a list of typical agricultural plants, so crops, vegetables, or other type of crops. And you see here the genome size is usually then given in picogram DNA or in megabase pairs. And you see there's a huge variation. So there are even much smaller plants with smaller genome size, but there are also plants with much bigger genome size. So genome size in Ploidy correlates within only within the same species. So that means if we have a diploid plant, for example, let's say Raphanus sativus, this has in a diploid plant 1.11 picogram DNA, and in a tetraploid plant, a tetraploid has then 4 pairs of chromosomes. It logically will have exactly double the amount of DNA than the diploid. So the 4C content would be 2.22 picogram DNA. Yes. But that means also that a tetraploid Raphanus will have very different DNA than, for example, a tetraploid Pisum sativum. Yes. So we can compare, that's what we should learn, genome size and ploidy only within one species. Among different species is not possible. Okay, so summarizing that ploidy analysis is a measure of the number of chromosomes. And what we usually use in order to make a ploidy visible is a DNA fluorochrome, which is called DAPI. So that's a fluorochrome which excites a blue light. So we stain the DNA and then we can excite the DNA in the nuclei with the UV light, and we emit then blue light and can measure the quantity of DNA with this DAPI fluorochrome. On the other side, genome size analysis, we prefer to use a different fluorochrome, which is called propydium iodide. This is staining the nuclei with a dye fluorescing red, and we excite that with a different type of laser. So, but why? What can be reasons to measure the ploidy level? Here we may look at different types of users in the industrial field or in the academic or institutional field. So in the industrial field, of course, there are typical applications in the quality control of plant material, which is derived, for example, from in vitro material, because in vitro material often tends to have artificial, spontaneous modifications of the ploidy level, and regeneration of plants from that material may show undesired variations in the ploidy level. And then, of course, the whole area of checking ploidy level within breeding programs, which involve modification of ploidy. So there are some examples I will come to on the next slide. Last but not least, the monitoring, for example, of the quality of produced seeds. So seeds either may have a specific ploidy level, for example, triploid, when we look on, let's say, for example, sugar beet or other crops, citrus plants, often triploid plants are desired, and when you have plant or seed material which is sold, then the quality control of the desired ploidy level is made. And then if we look to the more academic institutional field, of course, here it's the same quality control of plant material in vitro cultures, for example, or within experimental breeding, of course, modifications of the ploidy level within the breeding process can be done. So a doubling of the chromosome number, for example, or a reduction producing haploid plants or double haploid plants are examples. And then, which is more topic in the ecology plant, ecology checking the distribution of natural ploidy variations in different ecosystems. Okay, I said I would give some examples. I will not go through all the entire list, but pick up some very interesting topics. So the detection of haploids and dihaploids. So in the breeding process, it is often used a technique to produce haploid individuals, and from them to regenerate dihaploids. So first there's a reduction of a diploid set to a haploid set. So we have a pure genetic information only derived from one parent, and then reproducing these haploids to dihaploid. Then we have a pure material which has not a mixture of homologous chromosomes from different parents, but just from one parent, and that means that's very well defined material for breeding. Another topic is hybrid breeding. So if you want to use plants which are very closely related for breeding in order to combine new genes, then ploidy analysis can be very helpful to see what a breeding strategy delivers. So if individual is a cross between two different species or not. So that's also where flow cytometry can help. And then, of course, the tetraploid. Tetraploid is a doubling of the set of chromosomes, of the regular set of chromosomes, which, for example, can be derived by a colchicine, by a chemical treatment, or triploids, producing triploids. So first you produce tetraploids and then you reduce them to a triploid plant. That is also relevant, for example, for oysters or shrimps or other fishes or shellfishes. Yeah, genome size determination, in contrast, is more of interest for, let's say, academic and institutional use. So it's related to the ecology, so to determine a species specific DNA variation, or to taxonomy, to create genome sites for a specific species as species characteristics. But also for hybrid breeding, or for the effects of genotoxic substances, maybe to change if exposure to genotoxic substances is modifying the genome size. You see this, all these applications are more related to research interests. Okay. Now, who measures the ploidy level? Of course, in industry, that's clear for seed producers, which are producing seeds of maybe modified ploidy level, or in order to verify the absence of so-called aneuploid plants or seeds. That means aneuploid seeds have deletions or additions of chromosomes and may form plants which are not so nice. Or have other defects, then service labs which offer analysis to other customers in gene banking, Ploidy plays a role, and of course not only in plant but also breeders in aquaculture, for example in oyster or fish salmon breeding may have use of ploidy determination. Yeah, in research and academic uses, of course, agricultural and aquacultural universities or university departments, public agriculture or aquaculture research institutes, or institutional agricultural research institutes and public gene banks are also potential users of interest for ploidy determination. Okay. Yeah. Now I showed a bit what ploidy is and for what kind of applications in breeding and in research it helps. And now a few words to the process, how it is realized in our solution. Okay, I already mentioned in a slide before that we are using DNA dyes which are fluorescing in order to label the DNA in the nuclei. So in plants, of course, we don't have single cells, as, for example, if we look in animals, then in the blood or in other liquids, body liquids, we have single cells which are not formed in a tissue, but in plants usually not. So first we need to take a part of a tissue, usually a leaf, which is most easy accessible, and then we have to destruct the cells, disrupt them to release the nuclei so that we get in liquid suspension of nuclei. And when we have this suspension of nuclei, we can add our DNA dye. So the DNA dye enters the nuclei and is binding to the DNA. And finally, then, when we excite them and when we use this fluorochrome mentioned DAPI, then we use the UV light for excitation. And the extracted nuclei begin to shine in this light. And finally, we can measure them then in a flow cytometer in the analyzing device, for example, the Ploidy analyzer. So how this is done practically? Practically, we take a small petri dish, usually a plastic petri dish, 6 centimeters, for example. We put a so-called lysis buffer into the dish, just a few drops, and a small portion of leaf tissue. We can also use other types of tissues containing living cells with nuclei, but leaf usually, of course, is best accessible. And then we destruct the tissue by a sharp blade, by chopping the tissue, so that we get a mixture of tissue fragments and extracted cell content. This extracted material we pour through a disposable filter, so that all the fragments of the tissue are retained, and only the suspended material is ending up in a test tube here with our nuclei in suspension, and we add then a second buffer which is containing the fluorochrome, and in the following we get the ready-to-use solution with the stained nuclei, which we can directly put on the analyzer in order to measure them. So this whole process of sample preparation takes usually less than 2 minutes. And then we go to the analyzer and analyze that, and also that measurement takes less than 2 minutes. And at the end, we have a so-called DNA histogram, which is displaying our results. So on the DNA histogram, we have two axes. One is correlated to the DNA content or the ploidy level, and the y-axis is displaying the number of nuclei the analyzer has analyzed for a specific type of nuclei. So that means if we have identical chromosomes, identical nuclei in these cells, one identical ploidy level, for example, a diploid, then we will see one population of nuclei forming such, we call that peak, yes, in a DNA histogram. And the position on the x-axis is indicating the DNA content or the ploidy level. That's both the same in this case. And the y-axis, if we just get the sum of all measured nuclei, is the base, how many nuclei have been acquired in this measurement. It can happen that there is not only one major peak, but that there are also further smaller peaks, like here. You all know that during the cell cycle, so the division of cells, there is a so-called G2 phase, which has a double DNA content, and this can form smaller or even bigger secondary peaks. But important is always the major peak, which is indicating us the regular DNA content, in this example, for example, a 2C content. Okay, in the video before, you already saw that we offer different solutions for staining the nuclei. And there are two different basic processes or procedures. I described in the 2 slides before the so-called two-step procedure, where we have first the extraction buffer or lysing buffer, which takes care for the extraction of nuclei. So it deactivates enzymes, it helps to permeabilize the membranes, and it's also helping to open the DNA structure so that the fluorochrome has a better accessibility to the DNA for binding. The second buffer then is a so-called staining buffer, which takes care for the final labeling of the DNA of the nuclei. It contains the fluorochrome and change the chemical environment for to produce the perfect conditions for the fluorochrome. Next to this two-step procedure, there is also a one-step procedure available, and this is combining basically both functions of the two-step procedure in one buffer. So we have one buffer for nuclei extraction and nuclei staining. So that's from the, let's say, laboratory routine, is a bit work less. So it's easier and faster to produce samples with these one-step procedures, but often the results are not that good as with a dedicated two-step procedure. But as long as the results are acceptable, it's let's say it's a reducing workload and makes life in the lab a bit faster and easier. Okay. Now, one example, for example, let's look at Vitia faba, so a bean, vegetable. We see here two histograms, two DNA histograms, and I indicated with this red ellipse the major peaks indicating the ploidy level. Again, on the Y scale, the number of nuclei, on the X scale, the DNA content or ploidy level. So if we have a diploid plant, it gives us a peak at a certain position. In this case, this is 200. So the number here, that's just a relative number. It's no absolute number. It's 200. And if we look on the second measurement of a tetraploid plant, it gives us a peak at 400. And we see directly 200 is diploid, 400 is tetraploid in that case. Of course, with the change of the ploidy level from diploid to tetraploid, also the label, the quantity of DNA fluorochrome is double, because we have the double amount of DNA, because we have, instead of two sets of chromosomes, 4 sets. So that's doubling. And this we can directly see in the position on the DNA histogram. So it's a very easy transfer of information. When we know this one is diploid, we know the other one is tetraploid. By chance, if the first one would be haploid, so just one set of chromosome, then the second must be diploid, because it's still a doubling of the DNA. You see, it's just important to have a plant in order to standardize the scale of the measurement with a known ploidy level, and then we can just derive from that information the related ploidy level of all samples which have been analyzed. Okay. You see, here is the same process again. The first peak is the important peak for us. Yes, if there are smaller other peaks, they are not relevant, because they can be caused, as I said, by a G2 phase nuclei after replication of the DNA, or by a so-called nuclei endoreduplication. So that's a process which can happen in a tissue, that the DNA is replicated without subsequent division of the cell. So then there are these endoreduplicated nuclei which are forming also peaks. But the basic principle, of course, is always the same. So the first major peak is defining our ploidy level. The second peak is caused by secondary processes within the plant and is not relevant for our result interpretation. Yeah, so lesson to learn, one plant may produce not only one peak, there can be several peaks. Okay, yeah, going a bit into detail to calculate the Ploidy level. And here, the technique is usually to work with an external reference. So what is an external reference? We have here again our DNA histogram with the Ploidy level on the X scale and the number of nuclei on the Y scale. So we use a reference plant with a known Ploidy level in order to calibrate now this scale. So that means as a precondition, we need to have a plant of our species of interest. So that's important. It must be a species of interest. So if we investigate on banana, we need to have a reference for banana. If we are interested in, I don't know, sugar beet, we have to use a reference of sugar beet. This is related. Remember one of the first slides where I explained the difference between genome size and Ploidy level. So the genome size in different species is different, but the Ploidy level, of course, is always related to the certain genome size in a specific species. So now we have our reference plants and we measure it, and it gives us the first peak. Yes. So for example, we can influence the position of the peak by the instrument setup. We set the instrument in a way that it produces a peak at 100. And now we know we have a diploid reference plant. The peak at position 100 is equal to a plant which is diploid. And now subsequently, we just measure our test plants, and this test plant now is producing in a different measurement on a different DNA histogram a peak at a position 200. And of course, we already know 200 is a double of 100. 100 is correlated to a diploid plant, so this must be a tetraploid plant. Or it shows a peak at 50. 50 again is a half of 100. So the half of a diploid plant is a haploid plant. So this is supposed to be a haploid, and so on. So we just can then deduct from the position of our test plants the Ploidy level they have. This is based on a simple formula, which is dividing the peak position of the reference and the peak position of the sample, and we multiply that with the Ploidy of the reference, and then the correct result is coming out. So that's just the same calculation I did before when I explained the graph. Okay, for situations which might be a bit more complex, and this is the case, for example, if someone would like to determine the genome size, or, for example, aneuploidy, because when we do that, we want to be very precise, and we want to have a very small standard deviation in order to detect small differences in the genome size. Then we use a different principle. So we do not have an external reference, but we combine in one measurement a reference with a sample. So we prepare a sample using a leaf of our reference plant and a leaf of our sample plant. We call that process co-chopping. And in that case, we can produce, or we usually will produce, two different peaks. And we can then use a formula to calculate the ploidy or to calculate the DNA amount, which gives us, let's say, hints on small modifications of the DNA, for example, for aneuploidy detection. Okay, now you can imagine, in order to obtain nice results, reproducible results like this, which is a very nice and sharp peak, we should look a little bit into how to obtain that. Yes, very important is the tissue selection. So the tissue selection should be, or should respect that we do use only intact leaf tissue. So tissue like that, which is already senescent, with dead portions, very old, should not be used, or which is contaminated by pests, yes, by animals or fungi. Of course, all these components could or will have also nuclei which are mixed with our cells. And they should not grow very fast because they will have a mitotic activity, and then the G2 portion of cells is very big, and we may have also effects on the G1. So that means we should optimally use very young but final fully expanded tissues. So young leaves which are already completely developed, they should be intact and parasite and disease free. Okay, not only leaves can be used, so practically all plant material which is containing living cells with intact nuclei can be used. So this can be leaf petioles, for example, but it can also be a part of the stem, of course, only when it's still living. Parts of flowers or of seedlings, and of course also seeds, yes, or roots. They all would be potential tissues, but of course, usually the leaf material is the most accessible. And in order to have good results, some further considerations. The plants should be well supplied with water before, yes. If they are under drought stress, then the tissue is maybe not, has a very soft state, and it is different to extract, or difficult to extract nuclei from that. You destroy nuclei. So very well supplied with water supplied plants are good. In research, of course, or academic environments, it would be good to keep herbarium vouchers of these samples, or to store them in a different way, in order to have the possibility to look back on the material. And if there are any comparative studies planned, then the cultivation of the plants should, of course, follow the same conditions, that you don't have modifications in the conditions the plants are growing in. Now, sample storage, it's often addressed question, um, uh, because not always the capacity for analysis is there when there is a material which should be analyzed. So for a short-term storage, the moistened chamber is maybe the most popular. So you just can take a wet tissue paper in a plastic bag together with your leaf material. And then, of course, it's even better when you put that in a refrigerator. And then usually you can keep this material for a couple of days and you will not see differences in the result. Of course, when the material starts fouling, then it's not usable anymore. Now, for long-term storage, so months to years, you can freeze the material. It can be desiccated, but only for the fluorochrome DAPI, not PI. That's not working. Fixation in ethanol or other fixatives is possible, but the quality here in these type of fixations is not that good afterwards. So my favorites are the freezing and the desiccation, which is practically giving excellent results after longer storage. Okay, this slide shows a little bit a different process of sample preparation because we have customers which are doing really huge amounts of samples a day. And then this manual preparation and filtration of single samples is a bit time consuming and nasty. There is also, of course, a different way to prepare samples in multi-well plates. So you can collect leaf material by leaf punches, put them into a well plate with silica beads, for example, inside. Then the material is, and you add buffer, then the material is grinded in a ball mill like the Geno grinder or different devices. So there are different tissue lyser, for example, is another brand name. And after that, you just can pipette the extracted nuclei with a multi-pipette into a filter plate and add the staining buffer, and finally analyze that with the Cyflow Space auto-loading system and the Cyflow Space system, so that you have up to 96 in a 96 well plate samples prepared within this process takes you maybe 20 minutes. And you have a very fast result of big sample numbers. Okay. Now we know things about the sample preparation, but what about quality control? So we see here an exemplary DNA histogram. So very important is that the peaks are symmetrical, so that there are no shoulders and distorted peaks, for example, so that we can clearly define the center. Then, of course, the so-called coefficient of variation is important. The CV is measured in percent, and if a peak is wider, the CV is higher, and if a peak is more narrow, the CV is lower. So for Ploidy analysis, CV values of 6 to 7 are still acceptable. For genome size analysis, it should be smaller. But here, this is usually an indication when the CV is high that there is room to improve the protocol, maybe the shopping procedure, or change the buffer to a different buffer. So we offer a couple of different buffers which can be tested. Yeah, and of course, a certain number of nuclei should be acquired. So for Ploidy estimation, usually a small number is necessary only because the peak position essentially will not change. For genome size determination or aneuploidy, the number should be higher because here we are looking on smaller differences in the peak position. Yeah, we also offer a reference and calibration material for the instrument. So this is called DNA control for different dyes, for the DAPI dye and for the propydium iodide dye. We also offer calibration material for the instrument itself. What we do not offer is reference material for the plant DNA, because you can imagine there are so many different plant species on Earth, so several 100,000. And if we look to the crops which are of interest for breeding, there are still several hundreds. So these internal references for, let's say, a banana diploid or triploid reference must be sourced locally. That's not in our offer. Okay, yeah, just in the end of this section, few words about Ploidy analysis by flow cytometry. So I described the procedure and the total duration by a manual procedure to come to a result is definitely smaller than 3 minutes. Other alternative methods, for example, microscopic analysis or counting of chloroplasts for genome size determination, Folgen densitometry, is a very work intensive and takes really a long time. So practically to flow cytometric analysis, there's not really an alternative here. Okay, good. Briefly coming to our full portfolio for the DNA analysis and Ploidy and genome size analysis. I think you heard already a bit about that. So first we have, of course, our Ploidy analyzer, the instrument, which we usually support and recommend when people have, let's say, a small to medium-sized sample volume and prefer manual sample preparation. So with this one, you can analyze DAPI or DNA content, genome size. There are different versions for Ploidy analysis, for genome size analysis, and for both two together. So 3 versions of the Ploidy analyzer, a very nice, compact, handsome device. And when, let's say, your sample volume is higher, there's a cyclospace, which also analyzes the Ploidy or genome size or the combination of Ploidy and genome size. So you can do both. The big advantage here is that this machine can be combined with an auto-loading station for 96 well plates. And this gives a high throughput of up to 300 samples per hour. So practically speaking, we have customers measuring in a day about 1 1,000 to 1,200 samples with these instrument, and it has an automated batch analysis, so it gives automated reporting and data analysis, and plate views, so indicates in the plate on an Excel sheet the ploidy level of the sample or in tables. It also has report functions for aneuploidy detection, so you can find out individual sample which have higher or lower DNA level as the regular type. Okay, we have a bulk of different reagents. Cysteine UV ploidy is our one-step procedure. UV precise P and UV precise T is our two-step procedures. They are also available in a bulk format for many samples. Yes, so these are bigger bottles, and the Cysteine UV Ox Protect, which is a bit specific because it is dedicated for problematic samples which tend to oxidization of the nuclei. Okay, so these are our ploidy classics. As I mentioned, they are available for ploidy or for genome size analysis, and for plants which contain so-called secondary plant metabolites, which interfere with the binding of the fluorochrome to the DNA. So these can be colorants, like present, of course, in wine grapes, but they can also be, of course, present in leaf material, flavors or tannins, which are in the leaf, and of course all these material, when we release the nuclei by chopping, are mixing with our extracted nuclei, and the nuclei are not protected because the membranes are destroyed, and this can lead to oxidation or any other kind of negative influence of the analysis. This can lead, for example, to these shoulders in a peak, which I mentioned, or to an unstable peak position, which is changing by time, bad CV values, because the nuclei after preparation are just not stable and they slowly decay. Yeah, for this type of samples, we offer a dedicated buffer system, which is suited for the analysis of oxidation-sensitive sample. It's called Cysteine Ox Protect. Also, this one is available for ploidy analysis with DAPI and for genome size analysis with propidium iodide. It's a one-step process. So it's not complicated and fast also to use. Okay, if we summarize what we offer to customers interested in ploidy and genome size analysis is a dedicated set of instrumentation which is fitting to the individual user need. Yes, and yeah, there's no redundant hard or software in one instrument, so the user only pays what is needed. So if you just need a ploidy analysis with one analyzing parameter and one light source, you only get that, and you don't have additional hardware which is inside your machine, but you never use that. And of course, we have ready-to-use dedicated reagents for ploidy and genome size analysis, and they are based really on a long experience. So I'm personally working 26 years in this field, but Sysmex Partec itself has looking back on 40 years of experience. And of course, everything is coming out of one hand. Okay, so I'm coming to the end of my presentation and just would like to give you briefly some very few exemplary applications. Yeah, one very popular is the production of seedless fruits. Yes, for example, watermelon or citrus or other crops. And this is usually reached by producing triploid plants. Triploid plants are sterile, therefore are not forming seeds, which is one yeah, very typical application. Or the doubling of chromosomes. Yes, doubling of chromosomes, that means can be from diploid to tetraploid or from tetraploid to octoploid and so on. Because the chromosome doubling affects many physiological parameters of the plants, so it can increase secondary metabolites which are desired, contained in the leaf material. It can make plants growing bigger or faster. So there are different advantages of just chromosome doubling. Polyploidy, so that means if we go to higher levels, again here we can enlarge plant organs. So to get the giga effect, for example, strawberries or other berries often are polyploid even on a natural scale. You can introduce or create higher yield or juice content or whatever compared to diploid, or just for ornamental crops to improve the size of a plant as breeding target. Yeah, and then of course there are also undesired modifications in the ploidy, and this is especially related to aneuploidy. So if there are deletions or additions of chromosomes that can lead to distorted plants with lower yield, or they are not growing at all after they are seeded. So that are non-desired effects or non-desired spontaneous change in the ploidy level, like in vitro plant generation, can be another aspect. Yeah, last but not least, I spoke a lot about plant breeding, but of course also in aquaculture. Ploidy is a topic. In oyster breeding, for example, modifications of the ploidy to produce bigger oysters, to produce sterile oysters, is very popular. But that's valid also for other seafood and also for fish breeding, especially when we go into hybrid breeding, for example, hybrids of salmon and trout. So also here, ploidy is of interest in general fish breeding. Okay, yeah, finally, a few, just summarizing benefits. So what we offer as a Sysmex company, a complete solution for the DNA determination out of one hand. So that's a fast and easy detection in ploidy by manual preparation in smaller than 3 minutes. When we use automated analysis, it's even faster. And we offer a complete workflow. So that means the workflow from the sample preparation through the measurement, finally to analyzing the data and reporting the data. We have dedicated reagents which are tailored for the application. So we offer not only one solution. If one solution is not working well, we have alternatives. We have also solutions for, let's say, small sample volumes and bigger volumes with higher quantity and bulk quantities. And we have the dedicated automation and reporting function. But next to those standardized reporting formats, we are also open and available to customize formats for reporting on the purpose of an individual customer. So we also fit for purpose here the reporting functions. Yeah, I mentioned already the 40 years of experience in plant DNA analysis in industry and research. And we support our products really by specialists which look back on a long history and experience. Last but not least, Sysmex Partec is ISO 13484 certified development, production, and distribution and service company. So we are absolutely in line with the ISO normative. Yeah. Just a last slide to summarize. We have the complete solution, and we are the only one in the market who deliver really the only complete solution for our users and customers. Yeah, so that was my presentation. And well, I thank you for attending and listening. And